Myself, Dr. Raktim Mukherjee, Assistant Professor and Head of the Department, Shri P.M. Patel Institute of PG Studies and Research in Science, affiliated to Sardar Patel University. My institution is accredited with NAC and has UGC 2F and 12B recognition. The title of my today's talk is some of the cytokines that mediate immune response. In this lecture, I shall give example of certain representative cytokines that mediate the immune responses. So some of the key cytokines produced by the innate immune responses. The first one is Interleukin 1. Interleukin 1 is produced by macrophages as well as epithelial cells. Interleukin 6, they are also produced by macrophages and endothelial cells, also few of the T cells. Endothelial cells are the epithelial cells of the blood vessels, the endothelial lining of the blood vessel. Interleukin 12 produced by macrophages and dendritic cells. Students, in three different areas we have come across the term macrophages. But let's understand or recap what is macrophages. Monocytes, when they travel to the tissues, they would be called as macrophages. Tumor necrosis factor alpha or TNF alpha produced by macrophages and natural killer cells, and also in case of certain T cells. Interferon alpha by dendritic cells and virally infected cells. Interferon beta exclusively by viral infected cells. From the broad outline of key cytokines produced by innate immune responses, it becomes pertinent that interferon alpha and interferon beta might have a prominent antiviral role, and this we shall discuss at length. When you talk about the type of cytokine, because this lecture is about the various types of cytokine, this is one of the prominent review paper that has come up in the Journal of Experimental Medicine. Inflammasome signaling and regulation of interleukin 1 family of cytokines. So this paper discusses specific interleukin 1 family of cytokines that are expressed by cells as cytosolic proforms, and these family of inflammasomes are regulated by interleukin 1 family of cytokines. It's interesting that to discuss the discovery of cytokines, interferon alpha and interferon type 1 was in fact identified in the year 1957 as a protein that interfered with viral replication. And the activity of interferon gamma, which is the sole member of interferon class 2 family, was described in 1965. This was also the first identified lymphocyte-derived mediator. Thereafter, macrophage migration inhibitory factor MIF was identified simultaneously in 1966 by John David and Barry Bloom. So this is how the subject of cytokines prospered. And you know that cytokines are different from hormones, whereas the classical hormones circulate in nanomolar concentration that usually vary by less than 1 order of magnitude. In contrast, there are certain cytokines such as interleukin 6 that circulate in picomolar concentration, and this can increase up to 1000 times during trauma or infection. So cytokines are widespread and widely distributed, and they can lead to inflammatory response. So that paper leads to our discussion on interleukin 1. So first let's understand what is the cell source of interleukin 1. There is a wide range of cell source for interleukin 1. The monocytes, the macrophages, endothelial cells, fibroblasts, neuronal cells, glial cells, keratinocytes, and epithelial cells. These are the various cell types that produce interleukin 1. I already discussed about monocyte macrophage system, that monocytes, when you travel to the tissues, they will be called as macrophages. When you talk about the interleukin 1 family, they are a group of 11 cytokines, and they play a central role in the regulation of immune and inflammatory response to infections or even to sterile insults. Interleukin 1, the studies began on pathogenesis of fever, and these studies were performed by Ellie Mencken and Paul Beeson in the year 1943 to 48, and this was done on the fever producing properties of proteins released from rabbit peritoneal exudate cells. Now, what are the effects of interleukin 1? Interleukin 1 produces a wide variety of effects on the differentiation and function of cells involved. It can lead to inflammatory response, and it can also lead to immune effector responses. Interleukin 1 is intensely produced by tissue macrophages, monocytes, fibroblasts, and dendritic cells, but it's also expressed by B lymphocytes and natural killer cells such as microglial cells and epithelial cells. Now, if I discuss the inflammatory and immune effector responses of interleukin 1, I would have to give example of the important part of the inflammatory response of the body against infection. because the cytokines increase the expression of adhesion molecules on endothelial cells, and this enables transmigration, also called as dipedasis, of immune component cells such as phagocytes, lymphocytes, and these cells travel to the site of infection. They also affect the activity of hypothalamus, but this part I would keep as a later part of the discussion when I discuss fever as an exclusive discussion on example of cytokine action. Interleukin 1 also affects the central nervous and endocrine system, and it's an endogenous pyrogen, and this is what I have reserved for my discussion on fever. When I talk about interferons, they are low molecular weight regulatory cytokines produced by certain eukaryotic cells in response to viral infection. They are species specific but virus non-specific. When I say they are species specific, each species produce a different type of interferon. When I say virus non-specific, this species specific interferon is not specific selectively to certain viruses. They are non-specific to viruses. It means irrespective of which species the interferons are produced, they would eventually be in a position to kill all types of viruses, that is non-specific for viruses. Now, what are the classes of interferons? Interferon gamma is a family of 20 different molecules that can be synthesized by virus infected leukocytes, antigen stimulated T cells, and natural killer cells. That was about the interferon gamma family. Now when we come to interferon alpha beta family, they are derived from virus infected fibroblasts. Interestingly, interferons do not prevent virus entry into the host cell. Rather, the interferons prevent viral replication and assembly, and thereby it prevents further amplification of the virus infections. About interferon alpha beta, specifically when we talk about interferon 1 alpha, it's also called as a dual function cytokine. Why it is called as a dual function cytokine? It means it plays a role in the nucleus by affecting transcription, as well as it's an extracellular receptor mediated effects of classical cytokine. So therefore it is a dual function. Interleukin 1 alpha is synthesized as a precursor protein, and it is constitutively stored in the cytoplasm of cells of mesenchymal origin and also in the epithelial cells. Whereas in contrast, the monocytes and macrophages do not contain preformed interleukin 1 alpha precursors, but instead they rely on de novo synthesis. Since we talked about antiviral properties, type 1 interferons has a very big potential against treatment of COVID-19. Type 1 interferons have broad antiviral activity both in vitro and certain clinical trials, some of the experiments in late 2019 and early 2020, because this paper, if you see, is very latest, as latest as 2020, that type 1 interferon can be a potential treatment for COVID-19. Cytokine storm I discussed earlier is one of the major feature why COVID-19 patients die, or later on they develop into multi-organ failure. Now we shall discuss the antiviral action of interferon, how interferon alpha and interferon 1 alpha and interferon 1 beta leads to antiviral action. Let's try to understand that. First of all, the virus enters the cell. Once the virus enters the cell, before that, this is an infected cell and this is a nearby cell. So virus enters. Once the virus enters, the virus will lead to activation of interferon gene. This activation of interferon gene, together with the assembly of viral nucleic acids and assembly of viruses, would take place. The interferon gene will lead to synthesis of interferons. Thus, the infected cell will release the much needed interferons. And once after assembly of viruses, the release takes place, that virus in turn will infect the neighboring cell. An already infected viral cell will secrete the interferons. This secreted interferon will act on the neighboring cell, because the neighboring cell will have interferon receptor, and all those interferon receptor will bind to the released interferons, and it will lead to signal activation of genes. The signal activation of genes will synthesize antiviral proteins. These antiviral proteins will not only block virus replication but will also degrade the virus nucleic acid. Before I come to this slide once again, I would like to highlight that this is from Abbas immunology. Virus infected cell produces type 1 interferon. This type 1 interferon acts on both virus infected cell as well as uninfected cell. Once it acts on uninfected cell, the interferon will bind to the interferon receptor. This will lead to induction of enzymes that will block viral replication, and thus in an uninfected cell, interferon will produce induction of antiviral state, whereas the same interferon, when it targets the virus infected cell, the virus infected cell will activate the CTL, the cytotoxic T lymphocytes, and this cytotoxic T lymphocytes will bring about programmed cell death or apoptosis. There will be increased expression of class 1 MHC molecules on infected cells, and this infected cell, after class 1 processing and presentation pathway, will be killed by the cytotoxic T lymphocytes. So that leads to the antiviral action. I would just like to highlight, apart from the figure that I explained, that there are two important enzymes that render the cells resistant to virus infection, and those two enzymes often asked in MCQs is oligo A synthetase and special protein kinase. When an interferon stimulated cell is infected, the viral protein synthesis is inhibited by active endoribonuclease that will degrade the viral RNA. So someone asks you that it is known that in the interleukins or interferons modulate the antiviral activity, but how it modulates? Your answer should be that once interferon stimulated cell is infected, the viral protein synthesis is inhibited by active endoribonuclease that degrades the viral RNA. Apart from that, an active protein kinase phosphorylates and inactivates the initiation of factor ELF2, and this ELF2 is a transcription factor required for viral protein synthesis.